So today we begin the central dogma of molecular biology. We've talked about mitosis and meiosis. We've talked about Mendelian and non-Mendelian genetics. This is ultimately what makes it all possible. If it were up to me, this would be the first lesson of the course, because everything falls back on this central dogma. Now, I say that because I'm a geneticist, so to me, you start with the gene and you work your way up, and then you figure out how things work. To other people, they're reductionists. They're going to start at the organism and work their way backwards. So today we're starting at the very, very, very bottom. Not the very bottom. The very bottom would be atoms. But we're starting at the nuclear material and then moving upwards. There are three processes we're going to cover: replication, transcription, and translation. And for that last test, you know I'm going to ask you an essay question on each one of these. Replication is where you're going to copy your DNA. Transcription is where you're going to take that DNA and produce RNA. And translation is where you're moving the RNA into a polypeptide or a protein. Sure, I can say it again. No problem. Central dogma of molecular biology. Replication, you copy your DNA. Transcription, you take the DNA and you convert it to RNA, ribonucleic acid. And then translation is where you take the RNA, ribonucleic acid, and translate it into a polypeptide. Okay, replication, transcription, translation. I'll go through all three. Replication, copy the DNA, you replicate it. Transcription, you transcribe the DNA into RNA. Translation is where you're going to translate the RNA into a polypeptide. This is what ties it all together. Big picture. When do you replicate DNA? When does that happen?
Like in what part of my...
Sure.
Phase one.
No. Nice try. What? Synthesis phase, S phase. You copy it. All right? You have to prepare proteins. How do you do that? Well, you take the DNA. Where does DNA live? What's it made? DNA is the chromosomes. Chromosomes are made up of DNA and proteins bound around each other. Where do they live? In the nucleus. That's where mitosis comes into play. So these chromosomes are just super highly coiled up DNA. Now, from the last lecture about Mendelian genetics, what's a stretch of DNA called that encodes for a polypeptide? A gene. So you've got these chromosomes made up of DNA that are effectively sequences of genes. A gene encodes for a protein. So what's going to happen is this DNA gets transcribed into a piece of RNA. Okay? That RNA is going to move from the nucleus through what to get out of it? The nuclear pores. Goes out through the nuclear pore, and where does it go? Correct, the endoplasmic reticulum. So the RNA leaves the nucleus to the nuclear pore. It goes to the endoplasmic reticulum. What does it meet at the endoplasmic reticulum? So what makes the endoplasmic reticulum rough? Ribosomes. Meets a ribosome. The ribosome translates the RNA into a polypeptide. And then from there, the polypeptide is going to get some functional groups added, and then you'll get glycosylation, you'll get some proteolysis, and you end up with this long polypeptide. That polypeptide then starts to fold itself up. So you get the amino acids interact with each other. You form a primary structure, the polypeptide. Then you get secondary structures, alpha helices and beta sheets. Those then interact, and you get a tertiary structure, and then finally a quaternary structure, which has a final form and a final function. So what we're doing today is covering the part where you're going in the chromosome and the nucleus, where you're taking the DNA and you're transcribing it into RNA. So you're taking a piece of DNA and making a new piece of RNA that sort of matches up with it. That RNA message is going to leave and be translated at the ribosome into a polypeptide. That's all we're covering today. After today, you'll have the whole picture. Start to finish, you're going to start with building a protein, and you're going to end with that final protein product. So, back to nucleic acids, because I have a feeling you don't remember a lot of the stuff I talked about, 2 months ago. Good news, these are the exact same slides from 2 months ago. Literally the same thing. So if you've studied them before and you remember them, you're good. And if you don't, it may look new, but I promise you it's not. Nucleic acids are one of our 4 macromolecules. What are the other 3? So carbohydrates? Lipids? Those are, and proteins. Then you guys nailed the monomers of them. What? And nucleic acids is number 4. That's right. Yep. Good job. These nucleic acids are polymers. They're all going to be made up of a bunch of individual monomers called the nucleotide. Nucleotides look kind of neat. Here's their diagram. You've got a phosphate group, which is a functional group, a pentose sugar, pentose means 5-sided, and a nitrogenous base. That nitrogenous base may have 1 ring or 2 rings. So this is the general structure of the nucleotide. Comes into play because in DNA there are 4 different nucleotides. So talking about DNA and RNA today. DNA is deoxyribonucleic acid. RNA is ribonucleic acid. What's the difference? One is single-stranded, one is double-stranded. And that's what I'm going to teach you for Bio 101. Bio 102, we might cover triple-stranded DNA or double-stranded RNA. But for now, DNA is double-stranded, RNA is single-stranded. DNA is made up of the sugar deoxyribose, RNA is made up of the pentose sugar ribose. The other big difference is in the base pairing. DNA has 4 base pairs: adenine, thymine, cytosine, and guanine. So, adenine, thymine, cytosine, and guanine. RNA is going to be made up of a different set of base pairs, but very similar. You've got adenine, cytosine. This is DNA. Cytosine and guanine. But another one, this one has is one marked uracil. So it has uracil instead of thymine. These 4 bases are going to make up the genetic language. Here are the, in DNA, thymine, adenine, cytosine, and guanine. And you'll notice a few things about them. First off, thymine and cytosine are single rings. They're called, let's see if I can spell it right, pyrimidines. Adenine and guanine are termed purines. Adenine and guanine are the purines. Thymine and cytosine are pyrimidines. Here's how I remember it. I kid you not, to this day, this is how I remember it. The shorter word is the bigger structure. Purines? Oh, I guess now you have to figure out how to remember purines and pyrimidines. Okay, so here's how I remember purines and pyrimidines. Again, to this day, the same stupid mnemonic. I remember all, pure, and good. So adenine and guanine are purines. And then I remember the shorter word is the bigger structure.
But all the words are the same.
No. This purine is much shorter than pyrimidine.
So
Adenine and guanine are the purines.
oh, and those structures are bigger. Oh,
Right. Still not okay, Carly?
I'll figure it out.
Okay. Let me walk you through this. These two structures right here, okay? Thymine and cytosine are this very large word, pyrimidine. They are both pyrimidines. These two structures over here, adenine and guanine, are bigger structures, but they're a smaller word. Purine is both adenine and guanine. So, shorter word between these two words is a bigger structure. Now, the reason that it's so important to understand purines and pyrimidines as classes is because purines will only bind to pyrimidines. They don't bind to each other. So adenine always binds to thymine, and cytosine always binds to guanine. You guys are saying, this lecture is only 44 slides. It'll be over in a heartbeat. Lies, all lies. Here's why that's important. If I was to draw this out, we've got a phosphate group here, so phosphate, a pentose sugar, and a single ring. If it's a single ring, what is it, a purine or a pyrimidine? It's a pyrimidine. It's going to bind to a purine, double ring, like that. Does that make sense? So now you have double strand forming. And you can keep going and building it. This is the part where I get to look smart because I can draw a chemical formula. I'm sorry, chemical structures. If I knew what they were called, that would be even better. All right, so you can see by having 3 rings across, these form what are known as like the rungs of DNA. If you consider DNA a ladder that's twisted, these are the rungs. If you had a purine binding to a purine, how many rings would go across the center? 4, which makes one rung really wide. It would make it unstable. It doesn't happen. And if a pyrimidine bound to a pyrimidine, you'd end up with 2, so it'd be too thin. So you would end up with misshapen DNA that's just not stable in that configuration.
So they don't attach to themselves, they attach to each other.
Right. Pyrimidines will bind to purines. And they bind using what's called Chargaff's rule, otherwise known as the ATGC rule. Adenine binds to thymine, guanine binds to cytosine. So far, so good? Okay, now this may be the part you remember from high school. If I was to give you a strand of DNA, GATTACA, guanine binds to what? C. Adenine binds to? Thymine binds to? Thymine binds. There you go. So guanine. So we see the binding relationships between one strand of DNA and another.
You have a question like that?
You will. You will.
Really?
Yeah. It's a 6-point question.
Oh, it's an easy question.
Yeah. Well, then you have to do the next. No, you don't have to explain it either. You're going to have to go from a strand of DNA to RNA to a polypeptide.
And it can all be drawn.
Yeah. So far, so good, guys? Any questions? No questions. All right. So I think what's happened is, in the past, you guys have, it seems like you guys are really strong in this, matching letters to each other. But this is what I want you to understand, what's going on and why. So far, so good. Moving on. DNA is double-stranded. Each of these purines and pyrimidines is held together By hydrogen bonds. Now, are hydrogen bonds strong or weak? Individually. Individually, hydrogen bonds are super, super weak. I think I put it like, if a child was holding onto your hand, you can pull your hand away from them because you're a grown-up and stronger than they are. But if you have like 20 or 30 children holding onto you, you can't get them off of you. They're like leeches attached, and you can't do it. You have to like scrape them off. So DNA is very, very stable because of these hydrogen bonds. So the first thing we're going to do today is unzip the DNA. The first step is replicating it. Again, when does this occur? What phase of mitosis? S phase, right. Synthesis. Step one, you have to unzip this structure. Now, DNA has so many hydrogen bonds, you have to snap them. It's kind of cool. You don't have to understand this part, but I'll explain it because I think it's awesome. The way this happens is helicase, which is a protein, is going to come in and bend the DNA until it's stressed to the point that it snaps. Once it snaps, it becomes very easy to unzip. You have all had a book bag in your life where the zipper came undone, right? It broke, and then you couldn't fix it. It was really weak. That's what the helicase does. It comes in, and it snaps that zipper so that the whole thing starts to become undone. That creates what's known as a replication fork. So here's my shoe. That shoe's got too much of a knot. That one's better. All right, so DNA is double-stranded, right? So I got two shoelaces. There we go. I got stranded DNA. If I was to take that DNA and put a hole in it, open it up, this is what helicase does. It sort of splits the strands. So now you've got a fork. It splits. That's called the replication fork. Right, that fork. All right, we got a few important terms to understand. DNA polymerase, helicase, the leading strand, the lagging strand, the different nucleotides, we already covered them, and pyrimidines and purines. I should probably have moved this slide earlier in the lecture. So good news, we've talked about helicase, nucleotides, and pyrimidines. All we got left is polymerase and leading versus lagging strands. But if you were to hypothetically have to write an essay, these are the terms I'm looking for. Yeah, I did the same thing. I was like, All right. Here's an issue, though. The more I do this, if I was to set this shoe down and get these pulled tighter and tighter and tighter, they bunch up. They get tight here. Eventually, it gets so tight at this juncture that the strands will snap. So we have to relieve the pressure. In order to relieve the pressure of helicase opening up the strands, you have another molecule called topoisomerase that comes in. And that causes the DNA to spin. And that spin relieves pressure. So, step one, helicase will bind to the DNA. It begins to break the hydrogen bonds and unzip it. As it's unzipping, topoisomerase is right behind it, spinning the DNA to relieve pressure. All right? Once you've got that DNA strand unzipped, another protein comes along called DNA polymerase. Now, polymerase, what word does it have in it? Poly, and even more specifically, polymer. What's a polymer? Yeah, a whole bunch of monomers all strung together. What DNA polymerase does is it creates a DNA polymer. So it binds to the DNA after it's been opened up, starts running along it, adding nucleotides. Let me see if I can draw this out. Here we go. So we have a strand of DNA. Oh, I shouldn't have erased that. I could have kept it. G-A-T-T-A-C-A. And it's bound to its complementary strand on the other side, C-T-A-A-T-G-T. They're bound using hydrogen bonds. The DNA helicase comes in and binds, and it breaks that bond. Once it binds and breaks the bond, it starts to run the length of the DNA. As it does that, it's breaking more bonds. It has topoisomerase right in front of it, spinning the DNA to relieve pressure. The DNA polymerase will bind where that helicase was. And it's going to follow along the DNA. As it does so, it binds A to T, T to A, T to A, A to T. So you're making a complementary strand up here. Does that make sense? Yes?
Wouldn't that be RNA?
Say it again?
Isn't that the RNA that's...
Not yet. No, we're making a copy of the DNA first.
Oh, okay.
We're going to make RNA using RNA polymerase.
Okay.
We'll do that in a few minutes. So it's going to follow in one direction right behind the helicase. Problem is, and you'll notice on this image over here, the sugars are facing one direction over here, upwards. Sugars over here facing down. This is, let's say, this is 3'. I think this is 3' and 5'. So what's happening is the DNA polymerase can only follow the DNA in one direction. It follows the point of the sugars. Sugars are all pointing upwards. The DNA polymerase follows that. Do you understand what I'm saying? Here, the DNA sugars are pointing downwards. That's termed anti-parallel. They're sort of reverse of each other. So it has to follow it going down. So the other strand can't have polymerase bind there and move this way. It binds right behind the helicase and moves in the opposite direction. T, T, A, G. So far, so good? At some point, that helicase has moved farther down. Now you've opened up a bigger area. The top one can follow that helicase and keep making more pieces. One long strand, G, T. The bottom strand has to have another one bind out here and move backwards. So net result is you're getting the same DNA strand being built on either side. You're getting an exact copy created. But on the bottom one, you're going to have these shorter strands, what are known as lagging strands, because they're lagging behind. They're also known as Okazaki fragments after Drs. Okazaki. The top one is called the leading strand because it's following the lead of the helicase. So far, so good? Any questions? All right, so we've built two strands of DNA that are exactly alike. This is highly, highly conserved. Your DNA doesn't change much from one cell to the next. If it does, that's a problem. This replication is extremely accurate. There's less than one mistake in 1 million nucleotides. That being said, there are billions of nucleotides in your genome. Each time they have to get replicated in the billions of cells in your body. So mistakes do happen. Luckily, when a mistake happens, your body checks up on itself. That's the whole point of the checkpoints during mitosis. Check the DNA. So they're constantly the DNA is constantly re-examining itself. Proteins are sliding along it. And if they find a problem, a few things can happen. One is the proteins will be in there, will cut out a section of the DNA that's broken and replace it. Sort of, it's like repairing a pothole. You just take out a section, fill it in again. It's like when you're doing, inevitably, when you get a zipper stuck in something. What do? All right, so you pull it down, and you pull it up. Oh, you're stuck, so what do? You pull it back down, and back up, and back down, and back up, until eventually it works. That's literally what your DNA is doing. But here's the thing. What would happen if you couldn't fix the DNA, if the DNA stayed broken? Exactly, you go into G0. The cell stops reproduction. Now, if those checkpoints don't work at all, if the cell continues into replication because it cannot go into G0, what's a possible consequence? Cancer. Oh, it is? Growing old, cancer, and telomerase. Awesome. So here's the deal. This answers the age-old question of what do children and cancer have in common? Because that's something you ask all the time, I'm sure. Every time DNA replicates itself, your chromosomes are linear. They're a line. You get to the end of the chromosome. At the end, the DNA cannot copy the very, very tip. The tip just doesn't work. Because of the way polymerase is shaped, it sort of overhangs the tip a little. So you lose a little bit of your chromosome every single time you replicate. The older you get, the less of your original DNA you have. That's not a good thing. The reason that's so bad is because errors are going to begin to accumulate. The more errors you have, the more likely you are to get cancer. Children have very, very active telomerase. Telomerase is a protein that comes on after the chromosomes have been replicated, and it adds a bunch of random letters at the end. They don't do anything, but it acts like an aglet. Do you guys know what an aglet is? Yes. Because you watch Phineas and Ferb? Yeah, good, okay. Cool. So an aglet is the end of your shoelace, as we learned from Phineas and Ferb. It's that little plastic thing. It stops your shoelace from fraying. Telomerase acts to stick a piece of DNA on the end to stop the rest of the chromosome from coming undone. So it's an aglet. That's a great thing, because if that DNA came undone, your entire genetic code would fall apart. You would eventually, your chromosomes could not make proteins, and then you would die. Yeah. What? Why would that make you die if you can't make proteins? Proteins run pretty much everything in your body. Every metabolic reaction you have is mediated in some way by a protein. It makes those reactions that you need to survive happen. Without them, without the instructions to build those, you wouldn't be able to break down food, you wouldn't be able to build blood cells, you wouldn't be able to repair your body at all. Now, children have very, 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 very active telomerase. They have this thing working all the time. They are constantly fixing any damage that might occur. As you grow older, it doesn't work as well. Anybody ever broken a bone? Yeah, how long did it take to heal? A long time? 6 months? How old were you? 11? Oh, yeah, okay. Wrist has a lot of moving parts. And how long did it take to heal?
I was at a young age. I feel like it took like 2 months.
Yeah, it was quick. Okay, have you ever noticed, don't watch like old people fall over, but somebody who's older and they fall down, what happens? How long does it take for them to heal? Years and years, if at all. You had active telomerase. Your cells could reproduce and reproduce and reproduce and fix the damage. Their cells can't. That's the function of part of the function of telomerase.
So if telomerase fixes what's broken, right?
Right.
Why is it highly expressed in cancer?
Because cancer is brilliant at staying alive.
So it's just continuously trying to kill the cancer.
No, it's not trying to kill the cancer, because that's the cancerous cell. It has instructions for making more cancer cells, right?
Yeah.
So if that never breaks down, that cell doesn't have a finite lifespan anymore. Your cells have a little, this is a fuse for you. Eventually, it will get so short that the cell will go into G0 forever. Cancer cells are constantly rebuilding this, which means they will never enter G0, and they will never grow old. They will never experience the the failures of old age.
But they can eventually go into G0. Like if people,
Not cancer.
so people will always have it like in them, even if it's like dormant, I guess. Or
That's why you have to get rid of it. That's why you do radiation therapy. That's why you do chemotherapy. You have operations to remove it.
And that's why it's useful in killing cancer.
So there is one cell line. It's called the Helix cell line, named after Henrietta Lacks, who grew up in Halifax County, Virginia. Awesome. Henrietta Lacks was a woman who had cancer. She was an African American woman, and they took advantage of her like crazy. What happened was she went in to be treated for this cancer that she had. She knew she was dying. She went into a research institution. They said, we will take your cancerous cells and do research to see if we can help you. So they did. They took her cancer cells. They did research on them. She died. Her cells, though, are immortal. Her cells, her cancer cells, will live forever as long as they have nutrients being given to them, as long as they have food coming in. So they kept her cells alive. And her cell line became one that people would use for research all over the world. Yeah. They've found cures for lots of different diseases. They've created new medications based on her cells. And her family came forward, and they didn't realize that they had been using her cells. And her family said, why aren't we getting a cut of this? You're using, right, well, we're using this. But that's my mom. That's her legacy. We don't want you to use those anymore. And the research institution said, well, she signed this contract that said we had the right to any biological trash, effectively. She did sign it. The problem is she didn't know what she was signing. And this is where ethics comes into play. This is where she'd been cheated. And people made a whole lot of money off of her, and her family got nothing from it. Yes? Yeah, The Immortal Story of Henrietta Lacks. Yes?
I remember there's like an experiment where like in Harvard they grew the telomerase on like mice and stopped aging, so like would they still get cancer eventually?
It depends on the mutation rate. Eventually, if they continued and the mutation rate was high, yes, they would. But this would cut it down. As far as doing research into immortality, this is the place to start.
Like what about the jellyfish? I remember like you explained that once.
Yep, the jellyfish has also continuously active telomerase. It's not a question of can't get cancer, but it's a question of its cells can keep dividing without any. So there you go. In case you guys ever wanted to look into how to stop growing old, that's the place to look. Telomerase. Drink water. Thanks. Thanks. Go back to that. Going into transcription. With transcription, we're going to take the DNA template, so the gene on DNA, and convert it into a piece of RNA. And it's not even converting it so much as building a new piece of RNA off of that plan. That's the whole point. Take DNA, make a little piece of that DNA into, use that piece of DNA to create RNA. The big terms we need to know are promoter, terminator, sigma factor, polymerase, template, and transcription factors. So if we were to have a strand of DNA, a coding strand of DNA, that is a DNA strand that has a gene on it. And again, we'll start off with G, A, T, T, A, C, A. The other strand will say G binds to C, A to T, T to A, T to A, A to T, C to G, A to T. These are all bound by hydrogen bonds because this is DNA. The first step, you're going to, in prokaryotes, you're going to have, what's a prokaryote again? Do you guys remember? The simple ones, opposite of eukaryote. They don't have a nucleus. They're going to bind to the DNA. It's usually right before the gene. Now this polymerase kind of looks like a bug. It's got a few pieces to it. The polymerase has two parts of it. One part of it is a helicase. What does the helicase do again? It breaks the zipper. And the other part's a polymerase. Creates a polymer. So you got the first part cuts the bonds. The second part spews out a new polymer. These little antennae are used for what's it called? They're used for sensing the environment. They're looking for what are called transcription factors. Transcription factors will speed up or slow down this process. Next lecture, we're going to talk a lot about how this is controlled. So, first stop, we have to bind. Sigma factor can bind really easily to DNA. The polymerase sitting on top of that sigma factor can't. It needs sigma factor to introduce it. Okay, so you guys have friends. We've talked about this before, right? Some of you do. I know most of you don't. Here's the deal. You have, if I'm not mistaken, a friend that absolutely relies on you to find them a date.
A date?
Yeah.
That's a joke.
Am I mistaken? You at least know somebody like this that cannot get a date for themselves. All right, so that polymerase cannot hook up with DNA all on its own. It doesn't know how. It needs sigma factor. Sigma factor is you. Sigma factor is the one that says, hey, maybe you two should get together. You bring them to each other, you get them together, and then that's that. Now, what do you do after you get these two people together? You regret it. You absolutely do regret it. But in that particular situation, after you brought them together, do you stick around? No. You get out. You make an excuse to leave. And that's what happens. So then sigma factor leaves. The RNA polymerase is now bound to the DNA. You're gone. You don't care what they do. In this case, what they're doing is that polymerase begins to unzip the DNA. It binds and starts to unzip it because it's got that helicase. It's going to run the length of the DNA, sliding along it. As it's sliding along, it's unzipping it and creating a new RNA molecule. So for here, now it looks like a mouse with antenna. Never mind. It's going to run along the DNA. A is going to bind to U for uracil. That's the big change. T to A, T to A to U, C to G, A to U. So you've created a strand of RNA at the top. That RNA is popping out of the back of the polymerase. If you look at the screen, you've got the polymerase running along, and you're binding A to U, T to A, T to A, T to A to U, C to G, C to G, C to G, A to U, T to A. You're making a long strand of RNA. Now you'll notice that the RNA looks suspiciously like the complementary strand of DNA. They are very, very similar to each other. In the same way that, again, back to your friends and dating people, you'll notice they pretty much date the same person or the same type of person over and over and over again. They don't learn. They don't think they're doing that. They swear it's totally different this time. It's not. So you've got roughly the same RNA bound to the DNA as the DNA that it used to be bound to. So now we have this RNA that's created. At the end of their very short relationship, they break up. The RNA, here you've got initiation. It binds. It's going to run along the DNA. As it's doing so, it's creating a piece of RNA. Then it reaches what's known as the terminator, or the stop point. That's where it's released. And they go their separate ways. 3 steps: initiation, elongation, termination. If you remember those 3 steps, you're fine. So initiation, elongation, termination. You initiate by binding to the DNA. The RNA elongates until eventually you reach termination, and it is let go. So at the end of the story, your DNA and RNA go their separate ways. They were never meant for each other anyway. The DNA zips itself back up. The hydrogen bonds reassert themselves. The RNA polymerase is ready to bind to another piece of DNA once you hook it up with it. And then you've got this piece of RNA left over. You've transcribed it. But now there's more, because this is just this long, huge piece of RNA. It has to be modified. To be modified, it's going to go to a place called the spliceosome. The spliceosome, get this, splices things. I know, right? Pardon? Okay. So, shoot, I wish I had some yarn or something.
You have your shoes?
I have my shoes. I could do that. All right, so at the spliceosome, I got one long piece of RNA. I'm going to splice this piece of RNA. So this is not going to work out well for me in the end. I could just use the board, but now I'm using my shoes. They're already off, so I may as well. So I have a piece here that's I've tied a knot. I've got a piece coming off the top. I can remove that. Now I've got a shorter strand. I can do it again until I have an even shorter strand. To show it on the board, which may be slightly easier, if I was to take this RNA molecule, put A, U, U, A, U, C, G, A, U, A. What I could do is remove a section of it, and then remove another section. Those sections I removed are called introns, otherwise known as intervening regions. They don't do much for the DNA. They have a few reasons to exist, but again, at this level, not much. They don't do much. Then I'm going to take these pieces that are left over. These are known as the expressed regions, or exons. And they get spliced together to form a much shorter piece of RNA. So far, so good? Introns are these pieces that are removed. Exons are the pieces that are expressed.
So the ones that are removed, you don't anyway?
Nah, we just get rid of them. Now, I say that for this level biology. The thing is, modern genetics has shown that they actually have an effect, and you can have alternate forms of splicing, but for this level, it doesn't matter. All right, but wait, there's more. We remove the introns, we splice the exons together. The next step is to add what's known as a poly-A tail, or a polyadenyl tail. Lots and lots of adenine on one side. A. And that acts as a fuse, because this, unlike DNA, doesn't have any hydrogen bonds holding it together. So you have this long A tail to, it's a fuse. What ends up happening is once it leaves the nucleus, these A's start to break off one at a time. And then eventually this degrades. It's coming unwound. It's like if you ever had a sweater that has that one little piece of not even a piece of lint, like it has a string, yeah. And you start pulling it, and then the sweater starts to come undone. Yeah, but you can't stop pulling at it because it's there. and then you try and take a pair of scissors and cut it, but you still know it's there and you still keep pulling it. It's like that. That's every sweater. So that's unwinding. On the other side, you get what's known as a methionine or guanosine cap. That cap is its golden ticket to leave the nucleus. So now you've got a way out of the nucleus. You've got a lifespan. This is called mRNA, messenger RNA. It can take this message out to the ribosome. So there we go, DNA versus RNA. Deoxyribonucleic acid and ribonucleic acid. We're taking the DNA and we're transcribing it into RNA. All right, so here's the deal. Now we've got the RNA. The RNA leaves through the nuclear pore, getting out of the nucleus. It's messenger RNA. It's been spliced down to a nice, smaller size. They can move into the rest of the cell, and that leads to a problem. Because in the nucleus, the cell speaks nucleic acid. The cell speaks nucleotide. Outside of the nucleus, the cell speaks amino acid. It speaks polypeptide. So we need to translate the RNA from a nucleotide signal to a polypeptide signal, or to a polypeptide. To do that, you need a translator. And the translator in this case is going to be the ribosome. So the ribosome consists of two pieces, the small ribosomal subunit and the large ribosomal subunit. The first thing to bind to the RNA is the small ribosomal subunit. That blue thing is a ribosome. So here the ribosome binds to the RNA. So it binds. It binds to a specific region. Once it binds, there's a signal that will attach to the RNA. That signal is made out of, go figure, RNA, and it's complementary to it. So here, if you have an AUC, the complement would be UAG. And it binds on there. That acts as a signal to recruit the large ribosomal subunit. So now the large ribosomal subunit comes down and binds. And it's got three parts to it. The A site, the P site, and the E site. So A, P, and E, otherwise known as aminoacyl, peptidyl, and exit. I know, it's really tricky that way. So the best way to do this is to sort of do a presentation. Could you come up here? Bring your chair. I'm going to grab this chair over here. Good. Okay, let's just put 3 chairs in a row right here. Yeah, 3 chairs right there. Get yours on the end. You have the A site, the P site, and over here, the E site. Okay, now I need you to stand up and come over here. And Gabby, can you come here too? Bless you. All right, so this is for you. This is for you. Get over there. So what ends up happening is we have another set of RNA called transfer RNA. Each of these students represents tRNA, transfer RNA. They're each carrying a peptide, an amino acid. So far, so good? Now, he's going to come over here and bind to the A site. Sit down. Good. It matches, his feet are going to match the RNA. So they sort of are complementary. They bind together like puzzle pieces fit together. Good? All right, once he's bound, it begins to sort of assembly line down. Now you move into the P site, and you come in, and you bind at the A site. So far, so good? All right, at the P site, you drop off your amino acid. Now you move over to the E site, you move over to the P site, and then the next peptide bonds. So now you drop off yours in the P site. Well, they actually attach because it's a polymer like that. You go out the E site. You shift over. I shift over. Okay, now you go through the E out. Apparently this is my amino acid. I bind my amino acid to there, and now we have a growing polypeptide. I go to the E site, and then I'm released. And it keeps going until you get a huge, long polypeptide. Your chair, yeah. The trick to now we figured out how this works. These ribosomes are usually hanging out down by the endoplasmic reticulum. And we figured out that there's a specific letter sequence to code for each amino acid. Now there are 20 different amino acids. Each amino acid might be coded for by more than one set of 3 nucleotides. So here are your big important terms. Codon, so start codon, stop codon, and reading frame. It's that codon that I want to talk about. Each and every amino acid is coded for by a series of 3 nucleotides. UUU, so uracil, uracil, and uracil, code for phenylalanine. Here, UUC. Yeah, I'm sorry. Let's go with UUA. UUA, so U, first letter, second letter, third letter A is leucine. What would AGC be? First letter A. Yeah, so A, G, C. Serine. It's okay. I will give you these tables. You will absolutely have these tables. So you could use either table. This is sort of the traditional one where you've got each codon listed and the particular amino acid that it binds to. This is one that I find preferable, where you start off in the center and work your way out. So if I was to say GCA, I could go GCA is alanine, or you could start GCA alanine. I actually, because this is how cool I am, I spent an entire afternoon printing this off and sticking it in a watch. And that's what I do in my office hours when you're not there. That's what I do. Yeah, it works. Now I can't really tell time on it anymore, but I can definitely easily reference the different amino acids according to their codon. Yeah. So you're going to have a string of these in a row. Usually, they start off with AUG in eukaryotes. AUG is both what's called the start codon, but it also encodes for methionine. You always start with that start codon. So if I was to give you a bit of RNA, you could translate it into a polypeptide. C, A, U, G, C, A, C, A, U, U, A, G. Oh, okay. We'll just not stop with that. U, A, A. Does that work? No, that doesn't work either. U, A, U, U, C, A, and then U, G, A. Can you create the polypeptide from that? We'll write it down, see if you can do this. So CAUG, CAC, AUU, AUU, CAU, GAGC. Yeah? You're good at it? You're good?
Am I good at what?
Can you convert that to?
Yeah, I could.
But I don't want to? All right, you want to do it together?
Yeah. C-A-U.
So you're trying to say, like, what is it val?
Yeah, what's the first codon? That is absolutely correct. Always start with AUG.
So the C before it.
That doesn't matter in the slightest.
So, but if it's already started, it becomes.
Right. But, and correct, if it's already started. So, what's CAC?
That's a hit.
Histidine, okay. His. AUU? AUU? AUG?
No, it's.
Oh, CAU, isn't it? GAG? Oh, well, shoot. Hold on. Okay, and then? UGA? Stop. So, that is the end. Stop. You finish. So your polypeptide is His, ILE, ILE, HIS, GLU. Okay? Feel comfortable with that? You sure?
That's like the answer.
That's it. That's the whole answer. That is the genetic code. You're going to use 3 nucleotides, a codon, in order to code for a particular amino acid. Didn't you do what? That's exactly right. What you're looking at is when we've created these codons, these groups of 3s, we've set up what's known as a reading frame. You can shift the reading frame one way or the other, and if you shift the reading frame, you'll find that the amino acid sequence changes dramatically.
So we shifted it to start,
Right, we shifted it to that AUG to begin with. Otherwise, you would have started off with what? Histidine? And then a whole bunch of random things. Take a look at this. So here are 3 sequences. These sequences are exactly alike. But what you'll notice is, if you start at the very beginning, you end up with 1 polypeptide sequence. If you skip 1 letter, you end up with a completely different one. And again, the third, a totally different amino acid sequence. Skipping 1 single nucleotide will lead to an entirely different polypeptide sequence. Yes?
So you said always start with AUG.
Always start with AUG.
but what if there is no AUG?
Then that's not a protein.
So are you like in the first one, we would skip the GUC and go straight to AUG?
Correct.
So
Yeah.
those first 3 just don't matter.
They don't matter at all. Always start with AUG. And then you stop when it hits the stop codon. Even if it's like start, stop, that's that.
So none of those have a stop.
Nope. Nor do they start at AUG. There.
Well, the first one can.
It can.
The first one. That's
Right.
valid.
So we've got these reading frames. If you were to skip one or delete an amino acid, add or remove a nucleotide, you'd end up with an entirely different reading frame. That's called a frame shift mutation. You've shifted the reading frame, so it changes the polypeptide dramatically. We have these specialized RNAs. This is what you guys were acting as when you were up here. These are tRNAs, or transfer RNA. You'll notice that it's complementary to itself. It binds to itself. It's like sticky tape. It's coming to be Christmas. You guys have wrapped presents before? Yes? And you know when there's a specific length of tape you need, because if you get it too long, it starts to touch itself. And then you can't undo it, and it gets really bothersome. Especially it happens with duct tape. Now, don't ask why I wrap Christmas presents with duct tape, but sometimes it's necessary. It holds really well. And then, okay, I'll tell you why. So it takes the kids like 10 minutes to open them, and that just extends Christmas. So tape will stick to itself. So does RNA. And when it sticks to itself, it can form structures. And those structures, those specific forms, will give it a function. In this case, you've got this anticodon at the bottom that will bind to the RNA in the amino acyl site. And then up there, that yellow piece is an amino acyl acceptor stem. That's where you're going to stick the amino acid. That's where the amino acid sort of binds to it. There are 20 kinds of tRNA. Why do there need to be 20 different tRNAs? Sorry, because you have 20 what? No. Because there are only 20 amino acids. They each have to carry their own amino acid. So, just like with transcription, there are 3 stages to this: initiation, elongation, termination. The same thing, again, because I'm trying to make it easy for you. Initiation, you get the start signal. You begin. Elongation, the tRNA is dropping off the peptide in the the peptidyl site, where you begin to grow a long polypeptide, and then you hit termination, where the stop codon is reached and the polypeptide is released. So initiate, you begin at the start. You get longer and longer and longer until you hit the stop codon when the polypeptide is released. And that's that. So you've got the polypeptide that was produced by the ribosome. It goes into the endoplasmic reticulum. You get some glycosylation there, so you're adding carbohydrates. It moves into the smooth endoplasmic reticulum, where you can get more functional groups added to it. From there, it gets shipped out to maybe the Golgi apparatus for proteolysis or glycosylation, maybe over to the lysosome, where it becomes an acid hydrolase, or to the peroxisome, where it becomes catalase. Maybe out to the membrane, where it becomes one of those integral or peripheral membrane-bound proteins. It reaches its location and then folds itself up to its final form, going through the hierarchy of structure: primary, secondary, tertiary, quaternary, until it has this final shape to it. And then, of course, the form of the protein will ultimately determine its function. So that's how it ties everything together. Sometimes there are problems, translational errors, mutations. One type of mutation is the frame shift mutation. The other type is called a base pair substitution. A base pair substitution is where you're going to simply replace one nucleotide with a different one. That's that. That will, in general, affect one amino acid. So you're not changing the polypeptide very much. If you don't change it very much, you're going to change the form just a little. So the form doesn't change much, so the function probably doesn't change much. This may help increase the functionality of the protein or decrease it. Usually, this is what a lot of evolution is based on, these single base pair substitutions. Additions and deletions, on the other hand, are going to have a major effect. You add one nucleotide. Suddenly, the reading frame is shifted. You have an entirely new set of amino acids. You add two nucleotides, sorry. When you add two nucleotides, the whole reading frame shifts again. Entirely new reading frame, entirely new polypeptide. What happens if you add 3 nucleotides? Well done. Yeah, you only change one, which leads to a much smaller change in the polypeptide sequence. In general, if you have a change in, if you add 1 or 2 nucleotides, it's going to have a major impact. If you add 3, 6, 9, it's not going to have so much. Because there's multiples of 3. Oh, okay. Most of the mutations that we know of that allow you to still live are single nucleotide polymorphisms. One small change at one nucleotide. Sickle cell anemia is caused by one change of one nucleotide at one position. And it will lead to blood cells that become sickle-shaped or shaped sort of like commas, meaning that they are more likely to clump together, leading to death, painful death. One change in one nucleotide can do that. Now remember, your cells are undergoing mitosis regularly. You have billions of nucleotides in your billions of cells, and if one of them goes wrong, then you can run into serious issues.
So you're saying that one is just changed, and when you say frame shift, you mean one has been added to it?
One has been added or removed. Yeah. So in order to help you study for this, I've created a handy-dandy slide comparing replication, transcription, and translation. But the big thing to note is I will expect you to be able to coherently describe the process of replication, transcription, and translation. Essay questions, 3 of them.
Oh, one for each?
One for each. Good news, good news. Transcription and translation, right? Initiation, elongation, termination. If you can fill in the blanks there, you're set. Questions? Content review. Do you have any questions about any of these?